Good evening. Welcome to the UCLA Mildred E. Mathias Botanical Garden Public Lecture Series. We're very pleased you could join us for today's presentation. The topic is Plant Stories Trapped in Tar, Paleobotany at the Brea Tar Pits, and I think you're really going to enjoy the presentations. I'm Victoria Sork, Director of the Garden and Distinguished Professor in the Department of Ecology and Evolutionary Biology. Many of you may not have visited the garden. It's comprised of 7 acres of plants that represent ecosystems around the world. We're also home to a wonderful herbarium that contains diverse collections for teaching and research, including excellent specimens from our Southern California region, and we also have a botanical library. Our mission is to promote botanical knowledge and appreciation of nature to the UCLA community and public at large. Our diverse collections are designed to assist the teaching and research missions of UCLA while creating a tranquil environment within the urban surroundings. I hope you will soon have an opportunity, if not for the first time, some of you for the umpteenth time, to visit our garden. We can't wait to open our gates to the public. Before this event begins, we want to acknowledge the Gabriana Tongva peoples as the traditional land caretakers of Tongva, which is the Los Angeles Basin and Southern Channel Islands. We pay our respects to the ancestors, elders, and relatives and relations past, present, and emerging of the Gabriana Tongva peoples. So let me introduce you to Ms. Jules Gooch, who's our amazing manager of public outreach and education at the Botanical Garden. She's coordinator of the public lecture series, and she will give you an introduction to today's program. Take it away, Jules.
Thank you, Victoria. Welcome, everyone. It's so great to have you all here tonight. I'm so excited to share this presentation with you. A couple quick things before we get started. Just a reminder to stay connected to the garden. On our website, you can sign up for our newsletter. That's the best way to find out about upcoming events. You can also find us on social media. We're on Twitter, Facebook, and Instagram at UCLA Botanical. That's the easiest way to find us. And as always, our education programs are free to the public. I imagine tonight we have people from all over the world. Feel free to say hello in the chat. We'd love to know who's here tonight. I'm also going to pop in a link if you can and you want to support our programming. This is where you can become a member and help sustain our free programming for everyone to enjoy. Also, I imagine we have some paleobotanists here tonight. I think I saw a few people excited on social media about this event. So if you're out there and you want to say hello, or put a little note about your research in there too. We'd love to know what other scientists we have here tonight. As Victoria said, we'd love to welcome you to the garden. We hope to be able to soon. And for those of you who have been here, you might know we have a part of our garden that we call the ancient forest. A little bit different than some botanical gardens. We're less focused on aesthetic displays. We're not full of huge beds of tulips or things like that. We very much are a teaching and research collection and also a beautiful space. And so a big focus of the teaching that we do here is about the evolutionary history of plants and how they relate to each other, how we have come to have this extraordinary diversity of plant life that we have on Earth today, and also maybe even looking back into the past at what led us to this place. So I'm very excited this evening to have the opportunity to focus in on Los Angeles. We have plants here from all over the world. Tonight, our speakers are going to be speaking to us about the sort of ancient ecosystems and flora of Los Angeles that we know about from the amazing Brea Tar Pits that are just up the street, just down Wilshire. You can go and see these extraordinary pits. So without further ado, I'm going to introduce our speakers. Tonight we have Dr. Reagan Dunn, who's Assistant Curator at the Brea Tar Pits. Thanks for joining us. And we also have Jesse George, who's a UCLA PhD candidate in geography. We're so excited to have you this evening. So I'm going to check, Jess, how's your, you want to try sharing your screen and we can check it? Do you want to go to
Sure. Let's try the PDF version.
And we have people from Ithaca, Aloha from Hawaii. We've got some people up on Tatavium land in Panorama City, all over California. Hello, everyone, and welcome. Let's see, we've got Maya, a museum educator at NHMLA and the Brea Tar Pits. Hello. This is wonderful to have everyone here tonight. Let's see. Thank you all for your patience. As you know, it's not always the easiest thing to deliver a virtual event.
Having some technical difficulties.
Yeah. Let's see if we can get that.
Well, should I try the PowerPoint really quick? See if that.
Sure, let's do that real fast. Oh my gosh, we have someone from southern Alberta, Canada. Lots of great classes up in the north. Right here at UCLA. Hello, Gordon. Oh my gosh, maybe the furthest away. We have a good morning from Transylvania. Hello, Tatar. Pine Mountain, Georgia. Okay, let's see, Jess. Let's try this.
Is that working? What are you seeing?
We're sort of seeing it smallish.
How fun. All right, so let's stick with that PDF option.
Okay, let's do it. We'll just make it as big as we can. I think people understand. Let's see. Oh, okay. We might have a further away than even Transylvania. We've got someone here from India tonight. Hello, Sisu. This is fantastic. Emily from the UCLA Geography Department. Hello. Okay, let's do this, Jess.
Okay. So, apologize for those technical difficulties. Actually, give me one moment. I'm so sorry, everyone. I don't know why this is reacting this way.
Yeah, someone just said, we've all been watching people wrestle with these presentations for a year. No sweat. This is a good reminder.
You had it down.
To other people when they're trying to bring you amazing free educational programming and teach you about the wonders of the world around you. So, I got you that.
Okay. Let's do it. Let's make it happen. All right. Is that okay?
You know what? It might be the best we have, and we're going to go for it. So,
I'm glad to hear it.
You're great.
And I'll believe you.
All right. So, Jessie George is a PhD candidate in the Department of Geography at UCLA. She has a Master's of Science in Environmental Archaeology from the Institute of Archaeology at University College London, where she studied ancient agriculture through preserved plant material from archaeological sites. So cool. Her current PhD research is on the fossil plants of the Brea Tar Pits, working to identify and radiocarbon date fossils in the collection to better understand how vegetation changed throughout time in Los Angeles Basin in response to changing climates and fauna. Her research interests in biogeography and archaeology are focused on the interactions of climate, plant life, and society. We're going to have Jessie present, and then we'll do a short Q&A. We'll have Reagan present, and then a little bit longer Q&A at the end. So take it away, Jessie. Round of applause.
Right, so hi, good evening. As Jules mentioned, my name is Jessie George, and I'm a PhD student here at UCLA in the Geography Department. My research is on Late Pleistocene vegetation and climate here in the Los Angeles Basin. I'll be sharing some of the work we've done so far with you. It's especially cool to be talking about this in association with the Botanical Garden here at UCLA, because it has a great representation of some of our modern flora. But if you go about 5 miles down Wilshire, from where the garden's located, we have one of the most exceptional resources for late Pleistocene ecology and a great record of some of California's past vegetation. So the Brea Tar Pits is known worldwide for being one of the richest ice age fossil deposits on the planet. Excavations have been ongoing at the Tar Pits location in present-day Hancock Park since about 1906, after the asphalt being excavated for Los Angeles's roads and rooftops was found to contain the fossils of large extinct mammals. Since then, millions of fossils have been unearthed and more excavated each day. The seeps responsible for this wealth of fossils originate from an oil-rich layer of Miocene rock, a part of the Monterey Formation, approximately 1,500 feet below the surface of La Brea. The oil in this reservoir is under great pressure. As a result, it travels through cracks and fissures. On its way to the surface, it loses its more volatile gases in that process. This crude form of oil is referred to as asphalt or tar. It forms sticky puddles on the surface, and depending on the surrounding ambient temperature, it can be sticky enough to trap the surrounding life forms to it. Everything from whole trees to giant mammoths to individual grains of pollen. So this asphalt preserves harder tissues extraordinarily well. So that's things like bone, chitin, calcium carbonate of shells, plant cellulose, which we'll be talking about, and allows for a really wonderful wide view of the ecosystems, Pleistocene ecosystems of the Los Angeles basin. Plants are represented in both macro and microfossils with their original tissue preserved. Macrofossil, when we're speaking specifically of plant fossils, is a fossil you can see without the aid of a microscope, something you can see with your naked eye. This includes leaves, cones, seeds, wood. We even have an entire tree preserved, something you might refer to as a megafossil. Microfossils, these are fossils you need a microscope for, include pollen, phytoliths, which are basically lithified plant cells, cuticles, which is the waxy epidermis of leaves, and spores of fungi. So it's extremely rare to find a site that so readily traps and preserves such a wide range of life forms over such a long period of time. We're talking 55,000 years, which means La Brea holds great potential for revealing not just subtleties of ecological and environmental interactions, but also how those interactions changed over time under changing climates. Plants are a great way to look at climate in the past. This is in large part due to the fact that they can't move, at least not in the way that we do. They have to be adapted to the surroundings that they're rooted in if they are going to be successful. We can use adaptations we see in modern plants, in modern environments, to reveal what environments were like in the past. By looking at the nearest living relative of a plant fossil, we can get more clarity on the conditions under which that fossil species was living in the past. We can learn about past conditions through how the environment influenced the chemical makeup of the plant through isotope analysis. This can give us valuable information on past precipitation and humidity. Leaf size and shape can reveal information on temperature and precipitation. Leaf fossils can also yield information on past concentrations of atmospheric CO2. So plant fossils have a lot to tell us, and we have a really long record of them. We have them preserved over a period of 55,000 years. This is important because it all falls within the range, one, of radiocarbon dating, giving us an important chronological context for each fossil. It also happens to be a particularly interesting period within Earth's history. The past 55,000 years witnessed the last extreme extent of glacier over the continent and the transition to our current warm interglacial. This is the period that saw the development of all of human civilization, in addition to the interruption of multiple abrupt, shorter, cold and warm events. It also marks the beginning of the sixth mass extinction. The late Pleistocene saw the disappearance of the majority of large-bodied mammals, megafauna. These are generally, mammals weighing over 40 kilograms. For La Brea, that includes mastodons, mammoths, giant ground sloths, and saber-tooth cats, among others. And understanding this has the potential to provide us with a deeper understanding of the environmental thresholds and resiliency of our terrestrial and marine ecosystems. Understanding the drivers behind these ecological turning points is an essential in a rapidly changing modern world. At La Brea, a little too fast, the mammals, especially the megafauna, have been the focus of studies for over 100 years, documenting extinctions, diets, competition between species. The plants, however, have been almost entirely neglected, with the exception of some valiant identification efforts made by the curator of the Los Angeles County Museum of Botany in the 1960s, Bonnie Templeton, and Janet Warder in the 1970s. Most recently, isotope analysis on juniper wood by Joy Ward and Lacey Gerhardt. There really hasn't been much done on the plants. These illustrations put together by Reagan, I think, really sum it up pretty well and give you an idea of the deficit in attention paid to plants compared to the animals. The Pits now only has 4,200 cataloged plant specimens compared to 540,000 cataloged animal specimens. And you can see how recently most of those cataloged specimens, or how recently most of those specimens were entered into the catalog here, sort of starting around 2008, 2009. And this in no way reflects the amount of plant material preserved. It's estimated that there are hundreds of thousands of already collected but neglected plant specimens at La Brea. Much of the increase in cataloging in recent years has been the result of research from this project and a group operating out of the University of Maine. But plants are an integral part to understanding changes in climate and extinctions we observe at the end of the Pleistocene. So what you're seeing this evening is really just the beginning of a lot of exciting stories and insights to come from the plants at La Brea. So hopefully just the tip of the iceberg. Our work with the plant macrofossils of La Brea is hoping to address some of the big questions left open by neglected plant fossils in the past. How plant diversity and composition responded to gradual and abrupt shifts in climate in the past. What do these plant communities tell us about local climate? Is there evidence for interactions between shifts in vegetation and faunal assemblages? So to answer these questions, we're identifying and placing the plant macrofossils at La Brea into chronological context using radiometric dating. The fossil deposits at the La Brea Tar Pits present some unique challenges. Chronological control is hard because of the mixing within the deposits, unlike more traditional excavation sites where fossils located on the same horizontal plane can be assumed to be from the same period. Fossils within the deposits at La Brea don't show clear organization. As you can see in this representation of fossils dates within one of La Brea's ongoing excavations, this is from Pit 91, time period is represented by color. Purples and blues are some of the oldest and reds the more recent. There really isn't a clear organization here. So in order to get around this, we have to date each fossil individually, which is exactly what we have been doing. So obviously we rely on radiocarbon dating for this study. This is a dating method that uses a radioactive or unstable carbon isotope as a measure of passing time after an organism dies. All living things contain carbon. That carbon is made up of 3 naturally occurring isotopes, carbon 12, 13, and 14. 14 is the only radioactive isotope of those 3 and is formed in the atmosphere when cosmic rays interact with nitrogen atoms. As plants take in carbon dioxide from the air as part of photosynthesis, they reach equilibrium with the constant ratios of carbon in the carbon isotopes in the atmosphere. As animals feed from those plants, and animals feed from those animals that feed from those plants, all life forms generally reach an equilibrium with the carbon isotopes in the atmosphere. Once they die and stop taking in that carbon, that unstable isotope of carbon starts to decay back into nitrogen at a known rate. And the ratio between carbon isotopes can tell us how long it's been since that organism was at equilibrium with the atmospheric carbon. So how long it's been since that organism was alive and taking in carbon. Because La Brea fossils are preserved in oil, a fossil fuel, we have an additional challenge of the carbon we're trying to date being preserved in an even older carbon, which has the danger of making our fossils appear older than they actually are. To protect against this, we very carefully remove any and all traces of asphalt from the fossils before dating, as you can see with the bleached product on the bottom right here. So you're seeing the journey of a cypress cone from excavation to the lab, Keck lab, where it will be dated. Yeah, and so for this study, the material was taken from the existing collections. Identifications on material previously identified were verified, and new identifications were made on materials that were excavated, processed in the lab, but never examined. Specimens were identified under a stereo microscope or fluorescence microscope using the Tar Pits reference collection, Rancho Santa Ana Herbarium, and Seed Bank, and the UCLA Herbarium as comparative collections. Thus far, we have identified and dated 145 new specimens. Here you can see the spread of, here are some pictures of some fossils and microscopes. And here you can see the spread of radiocarbon dates across the Late Pleistocene and Holocene. Each date plotted as a curve with standard error. Dates here are calibrated using the InCal calibration curve through OxCal, the latest iteration of global carbon-14 curve. And we can already see patterns emerging. The interglacial marine isotope stage 3, right at the beginning there, is very conifer heavy. We see pine, cypress, juniper, a lot of different species, and diversity drops pretty drastically mid-stage. So the loss of pine and cypress here already suggests possible changes to aridity. Through the glacial period, so right in the center there, you see MISQ, we really only see juniper species and some coast live oak sort of at the beginning, and coast live oak popping up at the end as well. So some really interesting patterns to note is the disappearance of juniper during the deglaciation. So as that glacier reduces in size and things are warming up, and what factors are behind the decline of a species that existed through the glacial maximum. This is a pattern that Reagan will be discussing further in her section and is pretty interesting. So just to give a little clarification on some of the patterns we're seeing here, I have some little time slices. So sort of early on there, right, we have all these conifers present. We have Pinus marcata, we have cypress, we have California juniper, we have bigberry manzanita, we have coast live oak, we have California dogwood. As we move into that glacial period, we really lose everything except for the junipers and coast live oak, and even the coast live oak really is just there at the beginning and the end. And then right at the end of that glacial period, we see diversity sort of expand once again. So what does this mean? Plants are a great indicator of climate because they are immobile and by necessity adapted to the conditions where they grow. Because of that, we're mostly dealing with species that are still present today. We can use the abiotic niche space occupied by modern individuals to draw conclusions about climate in the past. So this technique has its drawbacks. Species distribution models are most often created using modern species observations, which may not be indicative of the full suite of environmental conditions that species exist under. Species may exist under a wide range of conditions and therefore provide little precision when it comes to climate reconstruction. Here we use coexistence analysis to refine our climate reconstruction as a way of looking at overlapping abiotic niche space. So the overlapping sort of environmental envelopes that these species are existing in today. Climate reconstruction analysis uses coexistence likelihood estimation, or CRACL method, published by Harbert and Nixon. So for a given environmental variable, this method extracts distribution curves for each species present based on modern presences, and then identifies the best fit curve by calculating the maximum joint likelihood of coexistence. So for this study, we're looking at mean annual temperature and annual precipitation. So through CRACL and that coexistence likelihood analysis, we can get an idea of what's happening with mean annual precipitation and mean annual or annual precipitation and mean annual temperature. So you can see on top of our Planck chronology here, we have mean annual temperature in red here, and annual precipitation represented in blue. So variations here may seem more drastic and long-lasting than they actually are. So this is an artifact of the number of points where we're actually able to sample for the analysis. We're able to sample 9 points in time for species presences. So these are points that had more than one species overlapping. So do keep that in mind when looking at the patterns represented here. But we can see a trend of increasing aridity throughout marine isotope stage 3 and into stage 2. The glacial period indicated in blue, confirming the general picture we were getting from the species themselves, right? This increasing aridity. Temperature seems surprisingly stable throughout the record, with a couple deviations. This supports work done by Anna Holden in the past, looking at fossil beetles at La Brea. Accuracy of the coexistence analysis results likely suffers in the more recent interglacial as a result of much fewer fossil specimens dating to this period. And this may be the result of seep activity during this time. This could be how plants are interacting with the seeps, what species we have, are they more or less likely to preserve in asphalt? But for whatever reason, as of yet, we just don't have a lot of fossil representation from that more recent period. We do see 2 deviations in temperature, the dip at 38,000, possibly an indication of Heinrich stadial at this point, so a colder period that lasts approximately 2,000, and the rise at 11,000. So plant macrofossils alone are giving us an important dynamic picture of Los Angeles in the past 55,000 years. Surprisingly stable temperatures support earlier work, right? Looking at the beetles, we also see surprising patterns of aridity as Los Angeles moves from a conifer-dominant assemblage with species currently only found further north of the city to presences of only oak and juniper during the glacial period. There are many outstanding questions. More data is needed from the period following the last glacial into the Holocene to better understand the interactions between vegetation, climate, and fauna that occurred. But we at least have a start to establishing what was occurring at the base of the food chain. So now that I've given you a quick introduction to my work with the macrofossils and what we're learning just from the species presences that we can draw from these macrofossils and their radiocarbon dates, I think we'll take some questions, and then I'll pass you on to Reagan, who will share what we're learning from the microfossils. So thank you.
Thank you, Jessie. Or sorry.
So I'll stop sharing.
Okay. Great. Thank you so much. First of all, I'm going to pop up here just some general positivity. We do have a few questions in here. So yeah, it is exciting. It is wonderful. We do have a question here from Kendrick Welch. How do you find microfossils? Do you put all the dirt and tar excavated under a microscope?
That is a great question, and I think actually Reagan will be going over that process in her section of the talk. So I don't want to spoil everything, but it does involve a lot of dirt and tar.
Okay.
And microscopes.
And microscopes.
Fantastic. We'll look forward to that in the next talk. Let's see. Do we have some more? Oh, someone also wanted to know about La Brea woman, which I don't, it's not necessarily maybe part of your work, but they were referencing if that had any botanical associations with it, like some other archaeological remains of humans have had.
Yeah. It's not a part of my work, but as far as I know, she didn't, and I think what maybe was associated with her was maybe actually in the long run after dating, sort of a good example of mixing in the deposits, maybe not anywhere near her in time. So I don't think there were any plant material though associated with her.
That I remember that was something you described to me before, how the tar is so different from some just static rock formations, and that it really does have quite a stir of time in it as opposed to normal stratigraphy. Let's see. People are excited. Yay! Yay! Okay, one more question here. Can DNA from any of the specimens in the Brea Tar Pits be sequenced?
Great question. There's a lot of potential. We have a lot of original tissue. If you're talking about the animals, there's collagen that survives. So I think the thinking is that it should be, but attempts so far as I know, have been unsuccessful. So whether that's the actual preservation. And that material's just not surviving intact, or whether it's the interference of whatever's used to the solutions that are used to clean the fossils or extract asphalt from the fossils. So far there haven't been any successful attempts, but there's hope.
Great work, really cool, nice work. We have one more question here. Let's see, from Sisu. Does this study help find structural adaptations of plants using microfossils?
Yeah, I think so. That might be again a question for just purely looking at the macrofossils here. So I don't know if that's something that she'll go into. But as far as adaptations of the plants themselves, I think we are kind of assuming, given the proximity of time and the life history of a lot of the species that we're looking at, so the generational time, that there's not going to be too much difference in adaptations between the time periods we're looking at in the past and in their modern counterparts.
Wonderful. Okay, we've got a You Rock. Thanks for an excellent talk. What's the most interesting thing you found in your study? And then I think I see a few other questions that they're going to be covered in Reagan's talk. So I'm not skipping all your questions. We're going to be answered by a great scientist in a moment. So I'll let you wrap this one up, Jessie.
Yeah, sure. Oh, with the most exciting thing I found, I just giving dates to a lot of these taxa, that for a long time, we're just like, oh, these are plants that are present, but like have no, over that 55,000 year time frame, have no sense of where they fall and in relation to what changes in climate or what animals. So just having that timeline for it and seeing changes occur is just super exciting for me. Being able to see the flora of Los Angeles change over that period, I find pretty exciting. I love it.
We find it exciting too. I saw lots of clapping in the chat, so thank you again, Jessie, so much for your wonderful talk. And we are going to, let me get back to this chat. So we'll see you in a little bit, Jesse, for the end Q&A. And I've got Reagan here if you want to get your presentation ready. Let's see, Dr. Reagan Dunn is an assistant curator at Brea Tar Pits and Museum. She is a paleobotanist whose research seeks to understand the interplay between climate, plants, and animal evolution through time. She studies phytoliths, pollen, leaf fossils, and wood to reconstruct ancient vegetation structures and composition. She has a PhD in biology from the University of Washington, an MS in botany from the University of Wyoming, and a BS in biological sciences from Colorado State University. She recently completed 2 postdoctoral fellowships, one at the University of Wyoming and another at the Field Museum of Natural History, a favorite of mine if anyone's out there tonight. She has extensive field experience, having worked in the Western US and in Central and South America. She's worked for the National Park Service as a geologist and has extensive work experience in museums, including the Denver Museum of Nature and Science, Thomas Condon Paleontology Center in Kimberly, Oregon, the Burke Museum of Nature and Culture in Seattle, Washington, and the Field Museum in Chicago, Illinois. Her work at La Brea Tar Pits focuses on understanding vegetation and vegetation change over the last 50,000 years in the Los Angeles Basin. Take it away, Reagan.
All right. Thank you, Jules, for that. And Jesse, thank you for that most interesting introduction and great talk on your PhD work. I just want to give a shout out to Jesse that of those 4,200 plant specimens that we do have cataloged, she's responsible for most of those. So her work is very much appreciated. All right. Well, if you guys have ever been to the Tar Pits Museum, one thing that you might notice is that you have to look pretty hard to find any plants on display. So here is a picture on the right of some mammoths, and there's this little box over here that has a light on it where nobody's standing, and highlighted up here on the left is what's in that box. Currently, that's the only plant display that we have, aside from these big pieces of wood that were just sort of shoved under the legs of the megafauna. So we have a situation at the tar pits where the plants have been neglected for a very long time. And as Jesse showed us, the difference between the animals that are cataloged and the plants that are cataloged also has repercussions on how much we know about them, how many papers have been published. So over here is just a search on Web of Science for Rancho La Brea, and you can see that there's tons and tons of papers, over 400 on animals and various things. And right over here in plants, shown in green, there's so far 6 publications. And I was being pretty generous in considering what was a plant publication. So clearly there's a ton of work to do, and it's an open book, and we're so excited that we have this opportunity now. Because plants are key to ecosystems. They are the habitat in which we live. Plants control everything about the terrestrial ecosystem, including the water cycle, the greenhouse gas concentrations and carbon sinks, soil temperature, soil moisture, fire regime, and erosion. We know those all very well in California. Plant composition, animal habitat and diet, and the way that animals get around. And they're also the base of the food chain, right? So they are its primary productivity, the energy that everything depends on. So if you have some disruption to the plants, you're going to see those downstream effects that can have major ramifications for all of life. And of course, plants are affected by climate, and climate affects the kinds of plants that can grow and the adaptations that plants have for particular environments. And plants are really the recorders of past environments because of their ability to preserve those adaptations, both physical and chemical. So our research at the La Brea Tar Pits on plants, our main goals are to assemble an integrative paleo-environmental record for the La Brea deposits and the surrounding area to really understand the outstanding questions about environmental change over the last 50,000 years in our area. but also globally. It's a time of big change, as Jesse showed us. Some of the major questions that we have about this time period, I'm showing here a graph of data, temperature and carbon dioxide data from an ice core from Antarctica over the last 150,000 years. And you can see how temperature has gone through fluctuations. So long ago, there was a big warm up here that coincides with a big CO2 rise. You go into these more cool periods, the last major glaciation, and then this rapid warming event also that is accompanied by this CO2 rise. So there's a lot of interesting change there. So we want to know, how did habitat change with those CO2 changes, those climatic fluctuations, the glaciations and deglaciations, but also what's going on in this time period is the arrival of humans to North America. One of our ongoing questions right now that we're super interested in is climate and its effect on vegetation the cause of the megafaunal extinction that happened in the latest Pleistocene, where those megafauna died? Or were humans somehow involved? Or is there some combination of both factors going on? Also, after the extinction, we want to know how did getting rid of all these huge taxa, these landscape architects, how did that affect the vegetative landscape that we see today? And then there's all the other things, the little things that depend on plants. And so we want to understand how those changes in the plant populations and communities affected other communities like insects, birds, and amphibians and herp populations. And lastly, we really want to try to understand how the rates of climate change in the past, as seen in these ice core records and in our paleo archives, compare to the human-induced change that we are seeing today and what we're doing. And so just to put this into context, I wanted to add some data here showing where this graph left off in 1995. Carbon dioxide levels were 359 parts per million in the atmosphere. And today, just 25 years later, carbon dioxide levels are at 415. So you can see how much higher these values are today than they were even in the beginning of the Holocene, and certainly during the glaciation periods when it was very cold. And like, how are plants even surviving down there? But they did. Okay, I'm going to talk today a lot about plant microfossils. There were a lot of questions in the chat, and so hopefully I can clear a lot of those up. So how do we get plant microfossils out of the tar? And here's an example of, on the left, a block of asphaltic sediment that you can see is just enriched with the tar. It's really kind of gummy, it's sticky, it's pretty nasty with all that petroleum in it. So we use solvents to dissolve that away. And what you're left with is just the sediment itself. Now, the sediment can go through various treatments to pull out the kind of microfossil that you're interested in. And then we might mount those fossils, whatever they happen to be, on microscope slides and look at the tiny wee things on under high magnification. And so some of those things that we look at include pollen, phytoliths, and leaf cuticles. What fun. All right, so I'm going to kind of step through and examine what each of these kinds of fossils can tell us and what they tell us about ancient life at the tar pits. Okay, so what were the tar pits like during the ice ages? We know that there was a really diverse fauna of over 230 species of vertebrates, and those include an abundance of birds, mammals, lizards, etc. It's really the best known site in the world for these extinct Pleistocene animals, and especially the megafauna, those big guys that are over 40 to 45 kilograms. The tar pits are extremely rich in carnivores, and that's probably because the pits acted as a trap to entrap herbivores, and then The whole carnivore family would go and try to eat on the dying or dead herbivore and then get entrapped. So we see about 90% of the bones that come out of the pits are of carnivores. And of course, thanks to Jesse's hard work and others at the University of Maine, we have a growing list of plant taxa. And what we see are the dominants. Definitely probably the most common plant fossil we find are juniper seeds. You know how they're just everywhere and they can roll around, and they're really abundant in our record. But we also have oaks, oak leaves, acorns, etc., manzanita, pine, lots of compositae seeds, etc. And my question has been, what's the deal with the grasses? Where are the grasses? And in our collection, we only have 21 specimens. So that has gotten my interest, and I want to learn more. So how much grass was there at the tar pits? One of the best ways to study grass fossils is to look at phytoliths. And phytoliths are microscopic particles of plant silica that form in plant tissues. Phyto means plant, and lith means stone, so it's just literally plant stones. These form when silica is dissolved in groundwater. It travels through the roots, exits the plant through the stomata, and it leaves behind a liquid silica. Later, that silica crystallizes in sometimes specialized cells that plants make to accumulate silica, like this one here, this grass silica short cell, this little dumbbell. They can form in other cells, and they can form between cells. Lots of different plants make phytoliths. The grasses just happen to be really good at it. Phytoliths are not necessarily totally useful to identify plant species broadly, but within the grasses, they are very useful to get to some taxonomic identification. And these taxonomic IDs can provide some valuable ecological information. So here I'm showing on the right some examples of these specialized grass silica short cells and how they can be identified to the different groups. And up here in pink, we have the forest grasses like bamboos. And light yellow, we have these puid grasses. This is a subfamily that consists of the cool season grasses. These grow in high latitudes or high altitudes generally, cool environments. And they have what's called C3 photosynthesis. This other group, known as the packmads, consists of a large clade with 6 different subfamilies of grasses in it. And these are warm season grasses. They're typically drought adapted, and they most of these taxa have C4 photosynthesis. That will become important in a moment. So what do we know about the record of grasses and habitats from the past research that has been done at the tar pits? Well, one study in 1988 by Bill Ackerston sought to pull out these little vegetative boluses that were entrapped in herbivore teeth. So here is the little cavity. They dug out the plant remains from those cavities, looked at it under the microscope, and then classified them into broad categories. And those broad categories include grass, gymnosperm or conifer, and the dicots, the flowering plants. And what they found, they looked at 3 different species, and what they found is that bison mostly had a diet of woody dicot species, but they ate a little grass, and sometimes they enjoyed some conifer. Camels really enjoyed dicots, shrubby things like shrubby leaves you can imagine, and also a little bit of grass, about 10%. Horses, while they only had 1 sample, but horses seem to have had a much higher diet of grasses in there. There's another way to look at diet and what animals are eating, and this is by looking at the tiny little scratches on the surfaces of the teeth. And this is known as dental microwear. And this is work that has been done by Larissa DeSantis and her team. And what they do is they take an impression of the surface of the tooth and examine the scratches and pits. And those scratches and pits can tell you about what that animal is eating. And so they have done this work at La Brea and also found that the bison and the camels are mixed feeders, and the horses also sort of a mixed feeder, but ate more grass. So if you take just the animal interpretations, it seems like there's not a lot of grass around, perhaps, or are the animals just being selective? There's another way to look at the diets of animals and grass composition, and that's by looking at the stable isotope carbon-13 from tooth enamel. And so this can tell you what kinds of plants these animals are eating, either C3 plants like those C3 grasses, or C3 plants like all the other shrubs and bushes and conifers, or these C4 grasses. And these C4 grasses grow in drier and more open environments. And what the DeSantis group found is that most of the La Brea herbivores fall into this category over here. So in this range of the carbon isotopes. So really these animals are C3 browsers and or have a C3 and C4 mixed diet. So again, the interpretation seems that there might not be that much grass, or certainly C4 grass around. So I wanted to test this idea by looking for phytoliths. I've only been at the tar pits for a year and a half, and so much of that time has been during the pandemic when I haven't been able to go in and use the microscope and lab to process samples. But I do have a few samples that I would like to report on today. These samples come from two different sites. One here is our box 9 from Project 23. This is one of a series of 23 boxes that was exhumed from the parking garage at LACMA. This box happens to be about 45,000 years old, between 40, well, near 45,000 years old. And this other sample is from a salvage project. Here's a big clump of bone and asphaltic sediment that came out of the foundation of the new Academy Museum of Motion Pictures. And these samples are about 55,000 years old. And this is Greg Davies, our technician who was working on that site. Okay, so there is great preservation of phytoliths in asphalt. This is something we weren't sure about when I first got here, but it is the case that asphalt also preserves these silica phytoliths. And here are some examples of what we found in these samples. So these things are teeny tiny. Like you can line up 4 of these across one grain of, or one strand of your hair. These things are tiny. And they do tell us some things. So here's some examples of the different assemblages or different types of phytoliths we found. So I counted these samples to look at the percentage of grass versus woody elements. And that, just to give you an idea of how that is done, I count all the things that would occur in woody vegetation or closed environments, or in like shrublands, anything with its woody or non-grass. Those are called forest indicators. And then I compare the amount of those to those 2 groups of open habitat grasses. And so if we're looking at really grassy habitats, the majority of the phytoliths should be from these open habitat grass types. So here's what we found. Of our 2 samples, you can see that we have a lot of yellow here. Yellow is the total of grass phytoliths in those assemblages. So we have over 83% of the top, the younger assemblage is made up of grass with a little woody material built in. And then also we see a little bit less in the way of grass and more woody at the 55,000 year interval. But this is really our first evidence of how much grass there is on the landscape at La Brea, or how much there was. Now, to break this down a little further, what types of grasses did we find? Are these C3, these C3 puids, or C4 grasses? And what we see is that in this older locality, a majority of the grasses here are of the C3 puid variety, so that cool season grass. At this 45,000 year interval, we have about 6.5% of these C4 grasses coming in. This gray area were grasses that were unable to be classified. But there is a little C4, but not a whole lot. So these phytolith data differ from the herbivore tooth bolus data by quite a bit. But they really do agree pretty well with the isotope data that show that there are C3 grasses, C3 woody plants, and with, going towards a little bit of the C4 end. And also it agrees with the dental microwear. So grass is present, it's available, it's really available for the animals to eat it. So now that the animal ecologists can go and study why or why not they might be choosing not to eat grass. Okay, so our conclusion, just based on these few samples, is that grass really was a major component of vegetation at the tar pits. We don't know how that will change with the different climatic events, but that's something we really want to know. And then we'll also be looking to see how those changes in grass composition affected the faunal compositions at the same time. Okay, I'm going to move to a sadder topic, and this involves the late Pleistocene extinction. Okay, this is the extinction at the end of the last glacial period. And if you look down here, this is the end of the last glacial period. We have this big warming event. And at that same time, we lose about 75% of all the terrestrial mammals, large terrestrial mammals. They're just gone. So you can imagine the biodiversity of mammals would have been much, much higher during this time than it is now worldwide. Of course, these are the dates for the extinction when it occurred in North America. It's slightly different in different continents. But Rancho La Brea really is the best deposit in the world to look at this and to look at the reason, first of all, to look at when this extinction occurred. And then because we have a plant record and we can get isotopes, et cetera, we can understand why this extinction might have occurred. And the one reason that Rancho La Brea is the best is because we can radiocarbon date almost every specimen. And there's so many individuals trapped in the tar pits over time that we have a really big sample size that we can deal with. So that's fantastic. So what is the cause of the extinction? Was it climate? Was it overhunting by humans? Or was there some combination of the two? Down here on the left is a figure showing the different glacial and interglacial periods over the last 800,000 years. So every time you see a peak up, that's a warm period or an interglacial. So there's been several in the past. So here, the last one was about 120,000 years ago. But we didn't see these massive extinctions then. So what is different about this latest Pleistocene extinction? And this is where we turn to the pollen record. Pollen preserved really well in asphalt as well. Unfortunately, I haven't had a lot of time to look at the pollen, so I'm just showing you a picture of La Brea pollen to let you know that we have it. But I'm going to talk about pollen records from the region, okay? And some of the archives that we have from a little broader area to try to understand vegetation change at the end of the last glaciation. And to do that, there are several sites where several long archives have been pulled from. And so these are typically lakes or marine basins. And the two I'm going to talk about here are the closest ones to Los Angeles, which is Lake Elsinore. And then here is the Santa Barbara Basin, where there's a core from the bottom of the ocean. So just to give you an idea of why lakes and ocean basins are really great for these kinds of studies. Here's a picture of Lake Elsinore. It's gorgeous. I've never been there. I really want to go. But these lake basins are these natural traps for pollen and other vegetation that either fall into them for the air or flow into them from rivers. So you can go to a deep part of the lake, drill the mud at the bottom, and you can see all these different layers. And trapped in all those different layers are millions of pollen grains. This is a picture of a core from Clear Lake. It's not the actual Elsinore core, but I just wanted to show that to show what a core looks like. So from those layers, you can pull out samples, and in those samples, you can count hundreds of pollen grains from hundreds of samples. And Linda Hauser has done this work with colleagues, Matt Kirby and others over the years. And you end up with a figure that looks like this. And so on the top right, you can see here are the relative abundances at each sample site, at each little interval where they sampled, and the kinds of pollen and the abundances of them in that little sample. And so you have all this data, but how can you interpret a data, such rich data? And one way to do it is to take that data and use some statistical methods. And what's really great is that there are databases such as this one, the Neotoma Paleoecology Database, where these data exist and are housed. And you can go and download them and perform different statistical analyses and look at the data in a different way. So I've taken this pollen data from Lake Elsinore and plotted it using a multivariate technique known as NMDS, and that's just non-multidimensional scaling. And what this does is that it groups the pollen assemblages by differences in the abundances of those taxa. And you can see that they sort of form these groupings. And you can take the scores, so you can take the x-axis score and the y-axis score and plot them on time. And I've done that over here. And this in blue is the y-axis score. And this is just a way to look at how all that vegetation data all combined changes. And so if you see a directional change, something's going on there. And then you can try to figure out, well, why is it changing in that direction? Then you can assign the time periods, which these colors here match up with these colors over here, so you can kind of train the eye of where these are in time. These are thousands of years ago. And you can look at the pollen record in more detail, and what you see most strikingly, if you just plot the dominant taxa in the core at these times, is that juniper pollen here shown in blue matches the shape of this juniper pollen matches perfectly the shape of this NMDS axis 1 score. So the decline in juniper pollen during this time period is really what is changing the fauna or the flora overall. And then you have these increases in pine and oak, or increase in oak or in gray here around the same time that you have that decline in juniper. So something is going on, and it separates out in space. Okay, so what do we know about the climate at this time? Well, Sarah Feekins at USC and others have done some really great geochemical work to look at what's going on with climate, she among other researchers. And what they see is that there is a drying trend that starts about 15,500 years ago and continues up to about 9,000. So this entire time period where the juniper is declining, you have the climate is drying. The climate is also warming. So the juniper is dying off. It's being replaced by oak and chaparral vegetation. And so I was wondering, well, why is juniper so badly affected by this change in environment? And it turns out that juniper, the way that they regulate water through their stomata, the pores that release gas from the plants, take in carbon dioxide and release water vapor from a plant, juniper doesn't have any way to control those stomata. They just leave them open all the time, and juniper just sucks water out of the ground, and they don't care. They just go for gold. And so that makes them really susceptible to decrease in precipitation, but they're especially susceptible to decrease in winter precipitation. And so this indicates, and also is confirmed by some other modeling studies, that winter precipitation really declined during this interval. And of course, when you have decreased winter precipitation and warmer temperatures, what happens? Well, there's a lot of wildfire. So there's at the same time also increased wildfire activity. And so that really is driving the Holocene vegetation, shown here in red, to a different state, more towards the state that it is now. And it doesn't look like it's ever going to go back to the Pleistocene. And the megafaunal extinction occurs right there in this interval where all this change is going on. So the same is true in Santa Barbara, in the Santa Barbara Marine Corps. You also see a similar pattern, okay? This just happens to have a much longer record. And so here on the NMDS axis score, you can see the changing vegetation with the climate cycles. And juniper, or juniper, it's all related to juniper, is really responsive to these climatic changes. And again, what you see is that decline in juniper right at the critical interval, at the end of the glaciation into the Holocene in the extinction. And so this is pushing the flora to a totally new normal over here. And it doesn't look like we're ever going back to where we were in the Pleistocene. So just to conclude on that, the late Pleistocene extinction did occur during a time of pronounced climatic change. There was warming, drying, and increase in fire also as a result. The role of humans as a cause is still being evaluated. That's a tricky one to answer, but stay tuned. We're trying to find some ways to deal with that. And really the Holocene vegetation assemblages and what we have today are way different than the past interglacial vegetation. So this current interglacial is a lot different than the last one, and we're headed for even more change. And so just one last plug for the future of plants at the Brea Tar Pits. We're also looking to study these little leaf fragments or cuticles right here. You can see Connie picking some out in the lab, and we floated some out in excavations. And if you look at the surface of those leaf cuticles, you can see the leaf epidermis and the structure of the intel, and also understand what the structure of the vegetation was. And we'll be able to tell you how dense or how open the Brea Tar Pits were through these different climatic intervals. We also have a really great record of fossil wood that probably spans that whole 50,000 years. Not the same tree, different trees. Here's the team working on a log that was removed from Pit 91. And you can see that the preservation of the wood is exquisite. You can see all the growth rings. It's pretty much just like firewood off your firewood pile. And from these, we hope to get annual growth rates so we can look at changes in precipitation change. We can look at droughts and even CO2 starvation in the past. So very last, I just want to tell you that the tar pits have gone through a lot of iterations from way back in the Pleistocene into the Holocene, where the humans have had their way, drilling for oil and mining for fossils. In 1974, the Page Museum was built, and we can display all these amazing treasures. And moving forward, I don't know if you guys are aware, but we're planning to, we're hoping to work to get a new museum, or in addition to the museum at the tar pits, which we can feature a lot more of these plant stories. So please go to this website and you can see what we're planning, and there'll be a capital campaign coming out soon. So think of us. And with that, I'd love to take any questions.
Wonderful. Thank you, Reagan. I'm gonna bring Jesse in too, in case you want to reference different questions. All right, we have some good ones in here. I saw people getting their questions ready. We have one from Priya. Do the solvent and other treatments to separate the microfossils have the potential to affect the specimen's integrity or continued preservation?
Good question. I don't believe so. The specimens, once they are removed, at least for the microfossils, they seem stable. This has been a concern for some of the larger animal fossils, the bones, and our preparator, Stephanie Potts, has been working with different solvents, trying to find the best one for the best long-term conservation of the specimens. But as far as the microfossils go, we don't think that they're affected.
Let's see. We've got from Larissa. Awesome job, Reagan and Jesse. Have you found any or much evidence of Atroplex? Does its abundance change over time?
Larissa, excellent question. I don't know. I need to get some modern samples of Atroplex to see what the cuticles look like. It's possible. The cuticles might be identifiable. And also, I'd like to get some modern reference collections of the pollen. So check back with us in a while. But yes, it is, Atroplex is a C4 plant that is not a grass. So that's probably why Larissa is interested in that.
We've got one from Jewel. Does the amount of grasses reflect the existing diversity or just poor fossilization?
It could be a variety of things. It could be that the grass seeds weren't necessarily recognized. It could be that we have thousands of grass seeds sitting in those little envelopes that are in envelopes and bags and in boxes and drawers stuffed back in the collection that we need to go through. It's possible that they just weren't well preserved. So we'll be able to test that in more detail once we dig into the collection. Jessie, did you find many grasses when you've been going through?
There's some. I have a feeling they're just less likely, less hardier, less likely to preserve over time. But I do think with the focus that's happening, that there will be more. And we do, we have a couple of Atroplex too, but only a couple, and we can't date them. They're too small.
There you go, Larissa. Great.
Let's see. Got one. Do you see any evidence of plant community extinction opposed to plant species extinction?
Oh, very clever. Yeah, I think if you could consider, it depends on what you call a plant community. But if you look at those clusters of the NMDS plots, you might construe that. It's based on abundances. So that'd be a very interesting approach to sort of define those and see if those communities go extinct. Thank you, Chad.
This is a little one from earlier. Since I grew up in Thousand Oaks, this comes from Kristen, can you associate what you find in La Brea outside of LA correlated with those valley oak trees?
Jessie, do you see valley oak trees?
I have not. Of what's been identifiable in the macrofossils, I find coast live oak, which I believe is present in Thousand Oaks as well. So at least one of your main oaks is there. But I personally haven't come across any valley oaks. But again, if you think about leaf difference, at least between valley oaks and Quercus agrifolia, the coast live oak just has a much hardier leaf, right? Like much more likely to kind of stick around and preserve long enough to be covered in sediment and asphalt.
Also, we just got some love. Great work, everyone. This was fascinating. It's wonderful to learn more about this, and people want to know when you're going to publish, which is always the eternal question, right?
My boss wants to know that too. As soon as possible. I know that Jessie's finishing her dissertation in a year or so. But as soon as we get more data, but look for some publications on the extinction, by the end of the year would be great.
Wonderful. So we're gonna wrap up here in a moment, and I just want to say thank you again so much. I think I'm not from the Los Angeles area originally, and moving here, learning about the flora. And then when I first met you and started talking with you both and learning about how much of what we do have today was here during the Pleistocene, which was such a different time. And I know, Reagan, you mentioned, like, how are they even surviving these ice ages when we live in such a Mediterranean climate now? and then thinking forward to the future of what our climate will be like in Los Angeles and what plants will survive then. So lots to think about. But we have one last quick question snuck in. Any Engelmann oaks or scrub oak parts? Great talk, thanks. We mostly, did you say mostly Coast Live? That's what we found so far.
Coast Live oak has been the identifiable oak for me. Not to say that those aren't present and won't be found later on, but of those best preserved appear to be Coast Live oak.
And you bring up a great point, Jesse, that especially with this kind of work, with paleo work, we're just kind of always learning more information and getting a better sense of it and better picture. So I'm excited, as I think many people in the chat are, to keep seeing what you're pulling out of the tar. And I think we're all excited that there's a greater emphasis on plants now over at the Tar Pit. So thank you for bringing your expertise to the area. So let's hear it again. Let's hear some thanks for our wonderful speakers for spending their time with us this evening. Again, like I said, all of our educational programming is free. I'm going to put a link in the chat if you are able to, if you have that capacity to help support our programming. We would love your support. If not, keep coming back and enjoy all of these wonderful programs we're going to be having. So again, thank you so much, everyone, and good night for now. Take care, everyone all over the world. We'll see you next time.
Good night. Thank you all for joining.
Thanks.